Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator at Simulations Plus. I'll be hosting today's webinar behind the scenes, making sure we don't have any technical difficulties. In this webinar, Stephanie Kollmann, Scientific Product Specialist, will cover how to formulate dosing recommendations and identify optimal study size for biosimilarity trials using MonolixSuite. Using pegfilgrastim as a case study, they'll show you how they can utilize a published population PKPD model of the marketed reference product to simulate clinical scenarios for a hypothetical biosimilar. Scientific Product Enablement Director Geraldine Sellier will serve as our moderator today. A few housekeeping items before we begin. We take your privacy rights seriously by registering for and attending this event or participating in the Q&A session. You are allowing us to contact you for follow-up. You may ask questions via the Q&A panel at your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address all questions during the Q&A and after the presentation. And now I'll hand it over to Geraldine to introduce our speakers.
Hi everyone. So just a few words about Stephanie Kollmann, who will be our presenter today. So Stephanie earned her master's degree in mathematics from the University of Innsbruck in Austria. She then worked for several years as a biostatistician in clinical research at Assign DMB, and then also as a biostatistician consultant at L'Oréal in Paris. Throughout the years, she focused on planning and conducting statistical analysis and modeling in the pharma and the cosmetic industries. And finally, she joined the MonolixSuite product enablement team at Simulations Plus in 2023. So Stephanie, the floor is yours.
Thank you for the introduction, Geraldine. So welcome everyone, and thank you for joining. Today's webinar will explore how model-based approaches can be applied to support biosimilar development using pegfilgrastim as our case study. So biosimilar development brings together three key challenges. First, regulatory expectations keep evolving, with agencies now focusing more on model-based justifications. Second, technical complexity. Model-informed PKPD approaches help to evaluate how sensitive biosimilarity outcomes are to differences in key product or system parameters. And third, the cost of clinical trials, which can escalate quickly if the study design isn't optimized. In this webinar, you learn how current FDA and EMA recommendations support a model-informed approach, how to use sensitivity and power analysis to guide biosimilarity assessments, and how simulations can make study design both scientifically robust and cost efficient. So we'll start with the regulatory context, reviewing how the EMA and FDA define biosimilars and what needs to be demonstrated to establish biosimilarity. Then we'll move to the case study, beginning with the dose selection, where we identify which dosing level is most sensitive to potential differences between biosimilar and reference product. After that, we will look at study design optimizations, focusing on how to estimate the sample size required to achieve 80% probability of confirming biosimilarity. We'll then move to the main conclusions and discussion, summarizing how this workflow supports efficient, scientifically robust biosimilar development. And finally, we'll close with a practical example. So let's begin by clarifying what the biosimilar actually is and how it's defined by regulators. So the EMA defines a biosimilar as a biological medicinal product that contains a version of the active substance of an already authorized reference product, while the FDA defines it as a biological product that is highly similar to the reference product, notwithstanding minor differences in clinically inactive components. So in essence, both describe the same concept, a biologic that is highly similar to an approved reference. Here we see the core criteria used by both the EMA and FDA to define biosimilarity. So the EMA requires that similarity be demonstrated in terms of quality, biological activity, safety, and efficacy, while the FDA, on the other hand, states that there must be no clinically meaningful difference in safety, purity, and potency. The regulatory framework for biosimilar development follows a stepwise approach, meaning that evidence is generated progressively, each step addressing the uncertainties that remain from the previous step. So we always begin with analytical or quality comparability, where the molecular structure, physiochemical properties, and biological activity of the biosimilar are compared in detail to the reference. And this step forms the scientific foundation of biosimilarity. If analytical data show a high level of similarity, the next step is non-clinical or functional evidence. These studies, such as in vitro pharmacology or only when needed, in vivo toxicology, had verified that the observed analytical similarity translates into comparable biological function. And finally, we move to clinical evidence phase. Here, we confirm that both products behave the same in humans by evaluating PK/PD similarity, immunogenicity, and only if residual uncertainty remains, safety and efficacy. So once all these data layers are generated, regulators don't review them separately. EMA and FDA perform an integrated assessment, weighting the totality of evidence to decide whether the biosimilar and reference are truly comparable in overall performance. So for the EMA, the final evaluation is performed through a comparability exercise, which integrates all evidence, analytical, non-clinical, and clinical, to confirm that there is no relevant difference in quality, safety, or efficacy. Minor differences are acceptable if they are scientifically justified and shown not to impact clinical performance. The FDA follows the same scientific principle. Its decision is based on the totality of evidence, meaning that all data are evaluated collectively. The need for further studies depends on the residual uncertainty that remains after analytical and PKPD comparisons. So in both frameworks, this integrated assessment, whether called comparability exercise by EMA or totality of evidence by FDA, provides the overall conclusion on biosimilarity. So both regulators describe a biosimilarity assessment as a two-phase process. Here's how the two fit together. So the stepwise approach is about how the evidence is built. Analytical studies first, then non-clinical and clinical data, each stage designed to reduce uncertainty. The integrated assessment is how it's more about how the evidence is judged. So regulators bring all the data together to make one overall conclusion on biosimilarity. So EMA refers to this as the comparability exercise, and FDA calls it the totality of evidence, but scientifically they describe the same principle. Evidence is built stepwise and then integrated into a unified decision. So now the FDA has recently refined how this framework is applied. in October 2025, the agency released a draft update to the scientific considerations in demonstrating biosimilarity guidance, modernizing how evidence is weighted. So let's take a look at what's new in this 2025 draft. So in 2015 guidance, the 2015 guidance established a basic framework, a stepwise approach supported by the totality of evidence with comparative efficacy studies expected unless justified otherwise, and only a limited role for modeling and simulation. For context, comparative efficacy studies, so CES, are clinical trials that directly compare the biosimilar and its reference in patients to confirm they produce the same clinical outcome. The 2025 draft update keeps the stepwise approach, but introduces greater scientific flexibility. Now, comparative efficacy studies may not be needed when strong analytical and PKPD data already resolve residual uncertainty. So the guidance also encourages model-informed approaches and advanced analytics to demonstrate biosimilarity. And importantly, the focus shifts toward a comparative analytical assessment, here called CAA. They now described as the most sensitive test of similarity between a biosimilar and its reference. So comparative analytical assessment focuses on the laboratory and functional comparison, basically the lowest foundation step in the stepwise approach, testing structure, purity, and activity, and is now considered the most sensitive way to detect differences between products. So this update signals a very clear evolution, moving away from large confirmatory efficacy trials and towards a quantitative model-based demonstration of similarity. So we have just discussed what's required to demonstrate biosimilarity. Now let's contrast that with traditional generics. So generics are small chemically synthesized molecules. Their structure can be fully defined, so an identical copy of the reference product can be made. The manufacturing process is straightforward and reproducible, and regulatory approval relies mainly on bioequivalence. If AUC, Cmax, Tmax match, the generic is considered the same. Biosimilars, in contrast, are large, complex proteins produced in living cells. The structure and function depends on the manufacturing process, which means two products can never be completely identical. And because of this complexity, a simple bioequivalence study isn't enough. Regulators require this so-called comparability exercise. So while generics prove chemical identity, biosimilars must demonstrate functional and clinical similarity through a much broader stepwise scientific assessment. Okay, so now that we have covered the regulatory context and understand what agencies expect to demonstrate biosimilarity, let's move from theory to practice and look at a concrete example, pegfilgrastim case study. So pegfilgrastim is a pegylated recombinant human granulocyte colony stimulating factor, or GCSF. Pegfilgrastim is used to prevent and shorten episodes of chemotherapy-induced neutropenia, a dangerous drop in white blood cells that increases infection risk. It works by stimulating neutrophil production in the bone marrow, helping patients recover faster between chemotherapy cycles, reducing hospitalization. And because of its critical role and use in oncology, pegfilgrastim is a major target for biosimilar development, offering lower costs and broader access. Though demonstrating biosimilarity remains scientifically challenging due to the drug's molecular complexity. So let's imagine we are a company developing a biosimilar for pegfilgrastim. We have already completed the analytical and functional comparability exercise, demonstrating that our molecule matches the reference in structure, activity, and function. And now we are moving into the clinical stage, where the goal is to confirm PKPD similarity and rule out any clinically meaningful differences. To do that, we need to design a biosimilarity study. Specifically, we need to decide on the appropriate dose level and the number of subjects required to confidently demonstrate similarity. So now that we have defined our goal, designing a biosimilarity study for pegfilgrastim, let's look at what makes this particular molecule scientifically challenging. So pegfilgrastim displays a nonlinear target-mediated PKPD driven by neutrophil-dependent clearance. Because drug elimination and PD response are tightly linked, small changes in dose, potency, or clearance could affect exposure. And that's why model-based simulations are essential to assess how product differences might influence PK and PD similarity. The analyses in the webinar were inspired by two published studies that established the modeling framework for pegfilgrastim biosimilarity assessments. The first paper developed a mechanistic TMDD model with a quasi-equilibrium assumption. This model is built using 3 dose levels, 2, 4, and 6 milligram, allowing evaluation of dose-dependent nonlinearities. The second paper used a simplified TMDD model on a single dose level of 6 milligram, incorporating Michaelis-Menten kinetics. So that work focused on sensitivity analysis and clinical trial simulation-based power evaluation, providing the foundation for sample size optimization, which we'll discuss later. So for our analysis, we used the model from the first publication as the foundation for all simulations. And together, these studies provide both the mechanistic understanding and the quantitative tools that we applied in our case study. So now let's move to the first part of the analysis, selecting the optimal pegfilgrastim dose to detect potential PK and PD differences. So the first question we address is dose selection. Which dosing level provides the best chance to detect small differences between the biosimilar and the reference product. Using the published PKPD model, we evaluate how each dose level reacts to control changes in key parameters. In biosimilarity assessment, it's important to consider all major PK and PD determinants, such as in this concrete example, it would be potency differences. So potency differences reflected through changes in receptor binding affinity, which can alter target interaction. Differences in clearance arising from elimination pathways, pegylation, or formulation factors, which can affect systemic exposure. and deliver those differences driven by device or formulation variability, influencing not only the administered amount, but also potential bioavailability. And together, these three sources of variability determine how sensitive a study is to detecting potential differences in exposure and response. So now that we have outlined these parameters, let's step back and look at the structural model that describes pegfilgrastim's PKPD behavior. So pegfilgrastim's effects are tightly linked to both its pharmacokinetics and pharmacodynamics. So the way the drug is cleared from the body and how it simulates neutrophil production in response. The PD part captures neutrophil proliferation in the bone marrow and their maturation through transit compartments to finally lead to neutrophil circulating in plasma. On the membrane of the neutrophils, we have the receptor called granulocyte colony stimulating factor receptor. And this receptor will be the target of the drug. The PK part of the model represents absorption after subcutaneous dosing into the central compartment. And the drug can either be cleared through linear elimination or via binding to receptors on the membrane of circulating neutrophils. And this binding forms the drug receptor complex, which is internalized into the neutrophils and degraded. And we assume that the binding is very fast, leading to a quasi-equilibrium target-mediated drug disposition model. The fraction of a receptor which is bound to the drug is assumed to feed back into the PD part of the model by increasing either a proliferation and maturation of the neutrophils. So there is thus an interplay between the PK and the PD. The binding of the drug to its target will, via the feedback mechanism, increase the neutrophil count. More neutrophils means more target receptor, so more binding and more elimination of pegfilgrastim. And this will lead to a nonlinear PK and possibly to unintuitive behavior of the model. And in a biosimilarity trial, one would typically measure the free drug concentration for the PK and the absolute neutrophil count, ANC, as a PD biomarker. So now that we have seen how the model captures pegfilgrastim's PK and PD behavior, let's move on to how we used it, starting with the dose selection analysis. So to address the dose selection question, we simulated 3 dosing levels, 2, 4, and 6 milligram, presenting the range based in the original model. And the goal here was to understand how each dose responds to small changes in key parameters. So we ran simulations using a typical subject, meaning only the population parameters were considered without individual variability. For each dose, we calculated PK exposure metrics like AUC and Cmax, and PD response metrics, area under the effect curve, and ANC max, to evaluate how sensitive each endpoint is to parameter perturbations. For the simulation scenarios, we systematically varied 3 parameters to mimic realistic sources of differences. First, the delivered dose was modified from -10% to +10% in 2% increments, simulating potential variability in device performance for formulation delivery. Then we explored two additional parameters, potency here along the KD parameter, and clearance. But these were varied also all separately, not at the same time. So when we varied, for instance, KD, clearance and delivered dose, they stayed the same, and vice versa. So for each, we introduced controlled changes ranging from -50% to +50% in 10% steps. And this setup allowed us to isolate the effect of each source of variability and determine which dose level shows the highest sensitivity to detect product-related differences. This is a visualization of the workflow. We used a typical subject and simulated three fixed dose levels, 2, 4, and 6 milligram. And for each dose group, we explored different scenarios by varying one parameter at a time. So either delivered dose or clearance or potency. And the magnitude of change depended on the scenario. For each simulation, we calculated the PK and PD metrics and then expressed the results as a ratio relative to the reference. So let me show how the simulation scenario is set up in Simulx. So we can see how the scenario is structured. I simulate two groups in this specific case, a reference group representing the marketed product at the nominal 6 milligram dose, and the test group that includes a defined difference from the reference. So in this example, we are looking at the delivered dose difference of +2%, meaning that the test group receives an effective dose of 6.12 milligram. I simulate only one typical subject that is identical across both groups. And for the parameter element, I use the population parameter values coming from the publication, but without variability, meaning that all omegas are set to 0. As outputs, I select the smooth PK and PD concentration profiles. In addition, I also compute AUC and AUEC directly in the model. Let me show you how. Here you can see I basically integrate over the smooth concentration curves. Okay, going back to the simulation setup. Yeah, so also the original model includes a body weight effect on clearance and on volume. So for this scenario, I used the typical weight value for my simulated subject. As outcomes, let me collapse this. So as outcomes, I define both AUC, Cmax, AUEC, and ANC max. Yeah. So for example, ANC max is based on, well, derived from the smooth PD output vector by taking then the maximum value of this curve. And Cmax is calculated analogously, just based on the smooth PK curve. For AUC or AUC, I select also as an output vector the calculated AUC, respectively AUEC, and I will output the calculated AUC as a value. Even though I only have one simulated value per group, I choose as an endpoint the geometric mean as the summary statistics, and this allows me to compute then the ratio between test and reference in the group comparison, where I select the reference, so here the 6 milligram, as the comparator group. Finally, after running the simulations, I can see the outcomes summarized and directly compare the the test and the reference groups to assess any difference in PK and PD results. This workflow required in the interface multiple manual steps, but the workflow was actually optimized via the Lixoft connectors. Going back to the slides. Okay, so visualizing the results, we obtain a plot like this. So this plot shows how differences in delivered dose affect the AUC across 3 simulated dose levels. On the x-axis, we have the range of delivered dose differences from -10% to +10%. On the y-axis, we see the normalized AUC values, meaning the AUC at each dose difference derived by the divided by the AUC at the reference corresponding to 0% difference. The colors represent the 3 simulated dose groups, and you can also see 3 horizontal lines that help interpret the results. So the black dashed line at 1 marks the reference. That's where there's no difference in AUC compared to the baseline. So if we were to see points laying on this line, it would mean that the AUC at, let's say, at +10% delivered dose difference is identical to the AUC at the unchanged nominal dose group. And the dashed lines at 0.8 and 1.25 represent the standard bioequivalence limits. In a real study, the 90% confidence interval for the test to reference ratio would need to stay within these bounds to confirm equivalence. Okay, so all three dose level groups, 2, 4, and 6 milligram, show comparable sensitivity along PK exposure metrics, AUC and Cmax. Exposure do not increase proportionally with dose delivered difference. If I'm using a 6 milligram dose, so here on the blue line, the 6 milligram dose and the delivered dose for the biosimilar formulation is 10% higher than for the originator. A proportional increase would mean that the AUC ratio would lay around 1.1, so around here on the y-axis. But we find values above for AUC, 10% increase in 6 milligram. The AUC ratio lies around 1.25. So on the PD side, represented by AU area under the effect curve and ANC max, there's visually no effect. The ANC response remains flat despite these considered dose delivery variations. So overall, all 3 dosing groups respond similarly to variations in delivered dose. When we vary binding affinity or potency, the 2 milligram dose shows the highest sensitivity to KD changes, while at 6 milligram, at 6 milligram, the effect is much smaller in that considered deviation range from -50 to +50% difference. At low doses, the receptor-mediated binding dominates drug elimination. The PD remains unaffected across all dose groups, reinforcing that the ANC feedback mechanism sort of dampens pharmacodynamic variability, even when PK changes are evident. So when we look at clearance differences, the picture starts to change. Here, the 6 milligram dose clearly shows the greatest sensitivity to clearance variations, while the 2 milligram dose is much less responsive. So at this low dose, plasma concentrations are low, and the drug is primarily eliminated through the nonlinear target-mediated clearance pathway. So changes in linear clearance have little impact. On the other hand, a higher dose in nonlinear target-mediated clearance pathway begins to saturate, making total clearance more sensitive to any further changes. I will elaborate this point in a moment. This means that at 6 milligram, even modest differences in clearance can lead to more noticeable shifts in exposure. And again, the PD metrics stay stable, which reflects the feedback regulation of ANC. Even when PK changes, the PD system compensates effectively. So as an explanation for why the 2 milligram dose group is less sensitive to clearance differences and the 6 milligram dose shows the greatest sensitivity, we can see here. So this bar chart shows how the relative contribution of clearance pathways changes with dose. So at 2 milligram, almost the entire clearance is target mediated, around 89% of total clearance. So in this region, the drug concentration is well below the saturation point of receptors. So at low doses, the system is highly sensitive to KD differences, but relatively insensitive to changes in systemic clearance. So as the dose increases, the TMDD pathway begins to saturate. Then by 6 milligram, a linear clearance contributes more, around 26% of total clearance, and receptor-mediated clearance is less dominant. So in this near saturation region, changes in KD no longer have a large effect because most receptors are already occupied. Instead, differences in linear clearance have a stronger influence on drug exposure. Concluding this part of the analysis, we assessed how small perturbations in delivered dose, linear clearance, and potency affected PK and PD metrics across the 3 dose levels. First, looking at PD, the PD metrics AUEC and ANC max remain essentially insensitive to these parameter changes across groups. And that's consistent with the feedback between ANC and pegfilgrastim elimination. Looking at PK, changes in delivered dose are clearly reflected in the exposure metrics, AUC and Cmax. And this effect is visible at all dose levels. So if the biosimilar delivers slightly more or less drug than expected, PK will pick that up reliably. Next, for linear clearance, the change in linear clearance is most visible at the 6 milligram dose. So at that dose, the linear clearance contribution is no longer negligible. So small clearance differences translate into measurable changes into overall exposure. And that's exactly the kind of clinically relevant PK differences a biosimilarity study should be powered to detect, especially at the therapeutic dose. And finally, for potency, the change in potency is most visible at 2 milligram dose. In that lower dose region, target-mediated processes dominate, so potency-related differences have a stronger impact on exposure than they do at higher doses. So based on these, and because 6 milligram is the approved clinical dose for pegfilgrastim, we will continue with the 6 milligram dose level for the next step, clinical trial simulations, and sample size optimization. So now we move to the second part of the analysis, optimizing the study design by estimating the sample size required to achieve 80% probability to confirm biosimilarity. So the goal here is to determine how large a study must be to reliably demonstrate biosimilarity for pegfilgrastim, given realistic differences between a reference and the biosimilar. And again, we focus on the 3 key sources of variability: dose delivery, clearance, and potency. Using clinical trial simulations based on PKPD, on the PKPD model, we estimate the power of the bioequivalence test analysis under different parameter perturbation scenarios. And from this, we identify the minimum sample size that still reaches the typical 80% power threshold. So this step translates our mechanistic understanding into practical study design, ensuring the clinical trial is both efficient and adequately powered to confirm biosimilarity. For the simulations, we used a 2x2x2 crossover design, mimicking a typical biosimilarity study setup. Each subject receives both a reference and test product across 2 study periods. The test product included small controlled differences in key parameters. We simulated 200 replicates per scenario, varying sample size from 50 to 250 subjects. So for each run, we analyzed standard PK and PD metrics, AUC from 0 to infinity, and Cmax for PK, and AUEC based on ANC from 0 to T last, and ANC max for PD. We then tested the design under different realistic product difference scenarios. So for dose delivery, we varied the administered dose by up to plus 10%, representing formulation or device variability. For clearance and potency, we explored a broader range from 0 to 75% for clearance and 0 to 150% for potency relative to the reference. And this setup lets us quantify how those parameter shifts, how these shifts impact the probability of meeting the bioequivalence criteria. Each simulation replicate was evaluated using a standard bioequivalence analysis, checking if the 90% confidence interval of the geometric mean ratio falls within the 80 to 125% bioequivalence margins for each PK and PD metric. We then calculated the percentage of successful replicates per scenario and plotted that against the sample size, giving us the power curves, essentially showing how many subjects are needed to achieve our target power at 80%. To make the simulations realistic, we included inter-occasion variability, IOV. This captures differences that can occur within the same subject across dosing occasions. For example, changes in injection site, physiological state, or procedural factors. And the published model reported an IOV of about 10%, but for our analysis, we increased it to 20%. And this adjustment reflects greater variability expected in a real-world multi-center studies and provides a conservative estimate of study power. So we simulated six. So this is how the workflow looks like. This is a visualization of the workflow. We simulated 6 milligram dose using the population model with both inter-individual and inter-occasion variability. Each simulation was run with different replicate groups and sample sizes ranging from 50 to 225 subjects. And then for every scenario, we adjusted those delivery or clearance or potency, then calculated the PK and PD metrics, and then performed the bioequivalence analysis. And finally, we combined all these results to compute the power, the study power, determining the minimum sample size required for a robust biosimilarity conclusion. So with that set up in place, let me show you how this would look like in the Simulx interface. Oh. Okay. So this is how the simulation setup is structured. For a specific parameter difference scenario within a single sample size batch and one replicate. So we can see I simulate 25 subjects per simulation group. So in total, I would have 50 subjects. One group follows reference treatment, reference test treatment sequence, and the other the test reference sequence. And this treatment sequence is controlled via a covariate element. I'll show you in the definition tab how it looks like. So here I have a covariate element called RT that follows the sequence reference treatment, reference test. So I can see that a subject receives for the first occasion, which corresponds to the first study period, the reference treatment, and in the second study period, the test treatment. Analogously, for the element TR, so the first during the first study period, the subject obtains the test treatment, and in the second period, the reference treatment. Also note that the body weight is again included as a covariate, since the model from the literature incorporates a weight effect on clearance and volume. So in the simulation tab, the administered dose corresponds to 6 milligram. The dose delivery difference is implemented through a formulation covariate effect. So when the subject receives the test treatment, the dose delivery parameter, here this parameter corresponds now to the bioavailability parameter, is adjusted accordingly to describe a +2% increase in delivered dose. As output vectors, I select both PK and PD time profiles, including now residual variability, so residual noise, coming from the original model. Running this simulation scenario, I will obtain the simulation results, the simulated data, and some nice plots. Here, for instance, the output distribution plot. But what I would want to do now is I want to export this simulation, this results of this specific scenario, to PKanalix to then conduct my NCA and bioequivalence analysis. I've done that in run. So this is how the simulated dataset looks like, loaded in or exported to PKanalix. I have here my ID, study period, so my occasions, time, observations, observation ID, since I have PK and PD data, the administered amount, and here now I have my covariates, the body weight, the formulation, if it's reference or treatment, and then also here the simulation groups that basically represent the treatment sequence. I set up my NCA, my non-compartmental analysis. At administration type, I select extravascular. I will start with the observation type PK. I'll select linear up, log down. It's already selected as an integral method for AUC. And as parameters, I will now select AUC from 0 to infinity and Cmax, also for a subsequent bioequivalence analysis. Scrolling down to the bioequivalence setup, you can see that the linear model is mostly pre-filled automatically. The only thing, the only field, the only factor I need to specify manually is the sequence, which now corresponds to the simulation group column in my dataset. After selecting NCA and bioequivalence, I can run the calculations and estimations. I have done in a prepared run, and we can see here the bioequivalence results, indicating that bioequivalence for AUC 0 infinity and Cmax, and we can see that bioequivalence has not been met, which is also confirmed by the confidence intervals plot, where we can see that the confidence intervals, they overlap with the predefined bioequivalence margins. So because I would need to repeat this process across many different parameter perturbation scenarios, sample size batches, and replicates, I automated the workflow using the leaks of connectors, making it much faster and fully reproducible. Okay, so now we can go back to the slides to have a look at the results. Here we can see a plot where the study power is plotted against the the power of the clinical trial simulation is basically a function of sample size. Here we see as an example the AUC metric for the PK metric, the AUC from 0 to infinity. So on the y-axis, we have the number of subjects included in the simulated clinical trial. And on the y-axis, we see the power, meaning the percentage of simulated trials that successfully meet the bioequivalence criterion. The colored curves represent different levels of the tested parameters. So in this case, delivered dose difference. And the horizontal dashed line marks the target power of 80%, which is typically considered acceptable for study design evaluation. So we'll start with the interpretation of delivered dose difference results, where we examined how small variations up to plus 10% impact the ability to demonstrate PK and PD bioequivalence. So for the PK endpoints, both AUC and Cmax reach 80% power up to 150 to 200 subjects when dose delivery differs by up to 2%. At 2%, AUC remains slightly below 80% threshold with 150 subjects, while Cmax shows somewhat higher variability, requiring, you can see it here, requiring around 200 subjects. And once the delivered dose difference exceeds about 4%, so here the curve in green, it becomes very difficult to demonstrate similarity without in practically a large number of subjects. For the PD endpoints, AUEC and ANC max, the bioequivalence criteria were met across all scenarios, confirming that PD responses are not sensitive to these small variations in delivered dose. So overall, this demonstrates that our design provides adequate power for realistic dose deviations, with PK exposure metrics being the primary determinants of study sensitivity. Also note, some power curves look a bit bumpy. These can be smoothed out by increasing the number of replicates from 200 to 500, for example. Okay, so here we looked at the effect of clearance difference on power to demonstrate similarity. So for PK exposure metrics, AUC and Cmax, we again see that around 150 subjects provide 80% power, when the clearance difference is small, up to 10% relative to the reference. Beyond that, power stays significantly lower, reflecting the strong influence of clearance on overall exposure. For PD endpoints, AUEC and ANC max, the bioequivalence criteria were always met, indicating that small changes in clearance don't significantly affect the ANC response. And for potency differences, modeled as changes in receptor binding affinity, KD, we observed similar trends. So for AUC and Cmax, around 150 subjects are sufficient to achieve 80% power, when KD differences are up to 25%. However, larger differences in binding affinity, such as 50%, a 50% increase, would require a substantially larger sample, around 250 subjects, to maintain power. Also discussing PD. So the PD endpoints once again met the bioequivalence criteria across all scenarios. So taking together the results, the results confirm that the design with approximately 200 subjects provides a robust power for detecting relevant differences in exposure, while PD outcomes remain stable across perturbations. To summarize the second part of the analysis, we used clinical trial simulations to evaluate how sample size and parameter differences affect our ability to demonstrate PKPD biosimilarity. For the PK endpoints, AUC and Cmax, about 200 subjects were needed to reach 80% power, when dose delivery differences were within around 2%. For potency differences, around 150 subjects achieved 80% power for deviations up to 25%. And for clearance, roughly 150 subjects were sufficient for differences up to 10%. On the PD side, AUEC and ANC max met criteria across all scenarios, showing that PD is less sensitive to product-related differences. Overall, these results confirm that our optimized design, a 6 milligram 2x2x2 crossover study with about 200 subjects in total, provides sufficient power for realistic differences and aligns well with the findings reported in the published pegfilgrastim sensitivity study. Okay, so moving on now to the next part of today's webinar, Conclusion and discussions. To wrap up the analysis, we looked at both dose selection and study design optimization for pegfilgrastim using model-based simulations. In the first part, sensitivity was evaluated across dose delivery, clearance, and potency differences. We found that only the PK exposure metrics were sensitive to these changes, while PD responses remained stable. Based on those results, 6 mg was identified as the optimal dose to carry forward into the clinical trial simulations. So in the second part, we performed clinical trial simulations along the same parameters to evaluate the required sample size for confirming biosimilarity. And here again, PK exposure determined the sample size needs. About 200 subjects were sufficient to reach 80% power for small differences of up to 2% for dose delivery differences and up to 10% for parameter differences. So the PD endpoints consistently met bioequivalence, the bioequivalence criteria, confirming that they were not sensitive to these perturbations. And important, all these findings were obtained under the assumption of 20% interlocation variability, representing realistic within subject differences expected in a crossover biosimilarity study. To make sure our simulations give realistic predictions, we evaluated how different levels of IOV affect the study outcomes. The 20% IOV scenario represents what's typically observed in clinical data, capturing the higher uncertainty, uncertainty you'd expect under real-world conditions. The 15% IOV case, on the other hand, reflects a more controlled study environment, like a tightly managed single-center trial. And with lower IOV, we'd expect higher power and smaller required sample size, simply because there's less random variability influencing the results. So here we compared both 15% to 20% to see how much that change influences the power estimates in our simulations. The upper plot here shows the power curves for IOV of 15%, while the lower plots reflect the same plots that we have just seen, based on IOV of 20%. And you can see that the curves shift upward slightly. The power curves are steeper, indicating less dispersion and greater precision in PK exposure estimates. Both AUC and Simulx reach 80% power threshold with fewer subjects when IOV is lower. The PK metrics for IOV 15% crosses 80% power around 125 to 150 subjects, compared to around the 150 to 200 subjects at IOV 20%. Here we repeated the clearance simulations with a lower interlocation variability of 15%. So as you can see, both AUC and Simulx achieve 80% power threshold with fewer subjects, around 100 compared to roughly 150 to 200 subjects before, when IOV was 20%. And the required sample size to reach 80% power drops by around, for differences in potency, drops by about 25 subjects. And we can detect KD differences up to 25% for an inclusion of around 100 subjects. For larger deviations, we would still need 200 subjects, but overall, lower variability translates directly into higher power and more efficient study design. So we need a good IOV estimate to get good predictions. Concluding, the overestimating IOV can make your study unnecessarily large, while underestimating it risks ending up with an underpowered trial. The assumed IOV should ideally come from published POP-PK studies under comparable conditions, like same dose, route, population, model, study design, etc. If no suitable data exists, a small pilot crossover can be run to estimate it directly, and in any case, actually, it's good practice to test study robustness across a range of IOV values, say from 10 to 25%. Okay, so now let's look at an example where the simulations we ran have a practical application. So this practical example uses two complementary publications. The first paper is an analytical similarity assessment for, again, pegfilgrastim. In this paper, the authors compare proposed pegfilgrastim biosimilar against the reference product. And what we take from this study is that they report protein content differences reflecting potential differences in delivered dose. In our simulation workflow that provides a practical basis for implementing a delivered dose factor, or along the bioavailability parameter between test and reference. They also quantify receptor binding affinity and kinetics differences using receptor binding characterization that includes a KD parameter comparison. And that gives us a second mechanistically relevant perturbation to implement in the model. The other paper forms the clinical confirmation step. This is a phase 1 single dose of 6 milligram two-way crossover PKPD similarity study comparing pegfilgrastim to the reference product in healthy subjects. They demonstrate PK and PD similarity using 80 using the standard 80 to 125% equivalence margins with 90% respectively, 95% confidence interval for PK, respectively PD metrics. And we use the comparative analytical assessment results from paper 1 to plan our biosimilarity trial and then benchmark our predicted sample size against the published PKPD similarity study in paper 2. So here are the specific parameter differences extracted from the two papers and used as input for the simulations. For delivered dose, the first paper quantifies protein concentration by UV absorbance at 280 nanometer. They report 9.9 plus minus 0.3 milligram per milliliter for the biosimilar versus 10.0 plus minus 0.4 milligram per milliliter for the reference, which corresponds effectively to minus 1.0% difference, where I calculate biosimilar minus reference. For binding receptor affinity, They use surface plasmon resonance to estimate K on, K off, and KD. And the reported KD is 150 picomole per liter for the biosimilar versus 180 picomole per liter for the reference, giving a difference of minus 16.7%. And for clearance, the value here is taken from the clinical data set in the second paper, reported as a geometric mean based on PK summary statistics, resulting in a minus 1.8% difference. So ideally, this step would rely only on the CAA-derived inputs. Here we have to assume basically a preliminary PK data that were available to support clearance estimate for both products. So the goal is to determine the sample size required to achieve adequate power for PKPD bioequivalence given literature. Given the literature-derived test differences. The simulation setup is almost the same as in the previous shown analysis. So the design, again, is a 2x2x2 crossover study with sequence from reference to test and test to reference over 2 periods with a single 6 milligram subcontinuous dose per period. The test arm, the test treatment now differs only through the literature-derived parameter perturbations, differences. And now the parameter scenario is the key point of this example. So we fix in this example IOV to 15%, and we apply the 3 literature-derived parameter differences simultaneously now, so all at once. And we run 200 replicates in our simulation. We evaluate different sample size batches up to 250 and compute standard endpoints. Here for PK, PK metrics would be AUC from 0 to T last and Cmax. And PD metrics would be AUEC, also from 0 to T last, and ANC max. Well, again, per replicate, we conduct, again, a bioequivalence analysis, calculate the power, and plot the power against the sample size batch. So now, based on the given parameter differences applied simultaneously, all endpoints across 80% of power at around 113 subjects under the combined literature-based differences. So these are the results of our simulations based on our workflow and the model that we have used. In the second paper, the crossover PKPD similarity study was successful with 56 subjects per sequence group, so 112 subjects meeting bioequivalence for the same endpoints. We have AUC from 0 to T last, Cmax, and AUEC also from 0 to T last, and ANC max for PK with 90% confidence interval, and for PD with 95% confidence interval within, and the relative mean ratio lies within the predefined bioequivalence margins of 80 to 125%. And this is consistent with our simulations, which indicates that total sample size of the order of 113 subjects is sufficient to reach the target power under the literature-derived parameter differences. And this shows how you can apply the workflow in practice to decide about the adequate sample size of the biosimilarity trial based on shown differences between the biologic in development and the reference product obtained during the analytical assessment. Okay, so all of this work basically from the TMDD model implementation coming from the publication to the clinical trial simulations was performed entirely in MonolixSuite using Simulx and PKanalix. The workflow we used here was straightforward to implement, and it can easily be extended to more complex designs if required. And because it involves repetitive simulation loops, multiple sample size, batches, parameter perturbations, and simulation replicates, it can be efficiently automized using the leaks of connectors, and this makes the process seamless and reproducible from start to finish. And finally, also the full workflow and the materials of this webinar will be made available so they can be reused and adapted both as a scientific reference and as a practical starting point for future biosimilar studies. Before I close, if you'd like to learn more, you can find our upcoming workshops, webinars, and online courses on the Simulations Plus event page, including sessions adapted to different time zones and in-person workshops at conferences. Well, and to stay up to date with new features, tutorials, and scientific content, you can follow MonolixSuite on LinkedIn or subscribe to our newsletter. Well, thank you all for joining today's webinar. I hope you found it insightful, and well, I guess we can now move to potential questions.
Thank you, Stephanie. Before we jump into the Q&A, I did want to launch a quick poll, and it's just asking if someone's interested in having someone from our team follow up with you about MonolixSuite or biosimilar development, and I'll give everyone a couple seconds to answer that.
While we wait, feel free to type your questions in the Q&A section. For the moment, we have very few, but don't be shy, don't hesitate to ask, and we will answer those right after the poll.
Thank you, everyone. I'll go ahead and end it there. And we can now take questions, Geraldine. Thank you.
Okay, so let's start with a question from the audience. So the first question is about the choice we made regarding the difference in delivered dose between the ref and the biosimilar. So why didn't we consider a difference such that we would be at the extreme of the bioequivalence limit, so at the 80 or 125 limit? I mean, would it also make sense to consider a big difference to be at the limit of the bioequivalent interval? Do you want me to take this one?
Well, I have an idea, but you can go for it, yeah.
So it really depends what are the questions that we are looking at. The question we were looking at today was a question in terms of power of the study. So we are in the setup where the two formulations are biosimilar, and we would like to check how many individuals we need to include to prove it. Well, if we would have considered a large difference in delivered dose such that the true answer is actually that the two formulations are not biosimilar. So, for example, just at the limit of the bioequivalence range. In that case, we would have rather checked for type 1 error. So that would be a different question that would be meaningful too. But today, really, the focus was on power, really in a setup where actually we have a small difference And the true answer is that the two products are biosimilar, and we would like to prove this. But if we are interested in controlling type 1 error, in that case, putting ourselves at the limit of the bioequivalent interval would make sense. All right, let's move on to the next question. I see that some participants would like to reproduce this workflow on their own molecule. So would it be possible to share the R scripts, for instance?
Well, of course, yeah, we intend to share all the materials that we developed for the case study we showed here today. So yes, we can share the scripts.
So all this will be available on our MonolixSuite documentation page as a dedicated case study that we need to finalize, but will be available soon. I also see a question regarding the structural model that was used. So Stephanie, could you maybe remind us what exact model we used for pegfilgrastim?
Sure, I could go to the slide maybe.
Yeah.
Yeah. Mm-hmm. Do you see the slide?
Yes.
Okay, so what was the question precisely?
I suppose it was, given that there are several models in the literature, just maybe a small recap of the one used for the simulation.
The one used for the simulation is it's a TMDD model with quasi-equilibrium assumption. Basically having a one compartment model for the PK here with a subcontinuous dose administration with first order absorption, linear clearance, but also here. I'm sorry, shall I explain the whole scheme or?
No, I think maybe just remind us in which paper we can find more details.
I see. Okay, sorry. The paper is listed here. 1 second. It's the Lee et al. Paper. The first paper that gives basically the mechanistic foundation of our case study. You find, we have the source listed here. You find here the DOE, where you can find in the additional supplement, the additional material, the non-mem model.
Thanks. And actually, we have an additional question on the model. So if you can go back to the scheme. So it seems that actually the PD affects the PK. So can you maybe comment a bit on this?
Yes. This scheme. Okay. Maybe for the mechanistic explanation, the precise explanation, this would exceed my expertise. Maybe Geraldine, you want to comment on this in further detail?
So there is indeed kind of a feedback mechanism. So the drugs affect the PD, and then because of this binding mechanism, we also have the PD that indirectly, in a way, affects the PK, and it's actually this feedback mechanism that makes also the PD pretty stable and not so sensitive to changes in delivered dose, as we have seen in the plot that Stephanie has shown. All right. So going into the the setup of the simulation. Could you maybe explain us a little bit how the weight covariate is considered exactly?
Yes, so the weight covariate has been included via power law relationship on clearance and on volume, and has been normalized by a typical value of the population that we have set throughout the whole analysis to 70 kilogram.
And then when we simulate the trial data set, how do we generate weight values?
We generate by sampling along a log normal distribution, giving a typical value and standard deviation.
All right, thank you. I see no further questions in the Q&A. Just to give a bit more time to people, if someone has still a question, maybe I actually have a question. So we have now taken population parameter values from the literature. So how robust are the results to the uncertainty we have on the used population parameters? Could you maybe comment on that?
So basically we did a robustness check based on the parameters that on a parameter that we took from the literature. It was the IOV. We relaunched simulations under different IOV assumptions. So we did a comparison between 15 to 15% versus 20% IOV. And we showed the impact on the required sample size or power on the power curve, and it states the practical implication that the overestimating IOV inflates sample size, and underestimating IOV risks an underpowered trial. And reasonable practice is to test robustness across a range. For IOV specifically, we recommended 10 to 25%. IOV parameters should be grounded in published POP-PK literature under comparable conditions. And I think this would also be applicable for other parameters such as TMDD-related parameters. So key model driving parameters, the robustness is best addressed, again, by sensitivity analysis. So you can vary parameters within plausible margins, and then check if those or design recommendations would change. And well, that's a suitable approach to understand how conclusions would change or move when parameters change.
Okay, great. Thank you very much. We have no further open questions. Jasmine, do you want to say a few concluding remarks?
Yeah, I just wanted to let you know if you did have any other questions, our BD team can reach out to you. We invite you to visit our website to learn more about this topic. I put a couple links in the chat. You can also follow us on social media. This webinar has been recorded and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you, everyone.